Hello, everyone, and welcome to the Rosa Worldwide Webinar Series. My name is Mike Reed. I'm the Chief Scientist at Rosa, and I'd like to welcome you to today's webinar. Just to note that our speaker next month will be Emily Nieves from Delineate in Cambridge, Massachusetts on February 11th, so please mark that on your calendars. During today's webinar, we're going to present it as a recording, and the presenter will be live to answer questions at the end of the recording. If you'd like to submit questions, you can do so using the Q&A button at the bottom of your webinar screen. And we look forward to getting lots of questions from you. Our speaker today is Dr. Alvaro Ruiz-Martinez. He is a modeler at Rosa. He has 12 years of computational and modeling experience related to mechanical, chemical, and biological systems. At Rosa, he's developed models focused on immuno-oncology, pharmacokinetics, and pharmacodynamics. He received his Ph.D. in mechanical engineering from the University of California at San Diego and did his postdoctoral training at Johns Hopkins University. Welcome, Alvaro, and we look forward to your webinar. Heather, if you'd like to start it. Heather, I'm not getting the sound from that.
Apologies. Let me share the audio. Just give me 1 minute. Sorry.
Mm-hmm. Yeah, one moment, everybody.
Hello. Today we are going to talk about an adaptable PBPK QSP gene therapy model that Rosa has developed and has been applied to IV administration of AAV2 across different species. First, we'll start with an introduction. Then we'll do a brief scientific and technical background. Then we're going to show how we develop this PBPK QSP model. After that, we'll show some simulations and the data we use for mouse simulations, NHP, non-human primate, and some human simulations for predictive purposes, just qualitative simulations. And then we finalize with some conclusions. So Rosa, in collaboration with Sanofi, has developed the gene therapy PhysioPD platform. Gene therapy is a rapidly growing area of research in the pharmaceutical industry. But despite its therapeutic promise, gene therapy research faces significant challenges, such as safe and effective delivery of the payload, of the AAV payload, dose selections, because it's not easy to predict translation from animals to humans, and we normally have more animal data than human data. Then there are sometimes unwanted immune system responses. And of course, this gene therapy research it has a high cost of development and treatment. Recently, quantitative system pharmacology approaches, or QSP, have been used to that impact gene therapy and the efficiency of gene transduction. Now, because of that, Rosa has developed a generic fit-for-purpose platform for AAV that stands for adeno-associated virus. So gene therapy for this virus, where we use this virus. And this platform or model covers a wide variety of scenarios and can be adapted to non-viral gene therapies, for example, in LNPs or lipid nanoparticles. Now, we'll show a brief scientific and technical background. We'll start with the scientific. We can say that we analyze with this research about AAV biodistribution and transduction. We try to define the key processes and rate limiting steps of these processes. Now, AAV-based vectors are the most promising and commonly used vectors for in vivo gene therapy delivery, owing to their safety and ability to drive durable therapeutic gene expression. Transduction efficacy is affected by the AAV distribution and serotype, also the target tissue, the route of administration, and the animal model. Indeed, the same AAV can display different biodistribution patterns, tropism, and transduction rates depending on the species. In this figure on the bottom right, this picture captures the key processes and the rate limiting steps that have been analyzed in literature, such as binding to serum proteins after systemic AAV delivery, vascular escape, receptor binding, intracellular trafficking, or nuclear entry, uncoating, and genome conversion that happens inside the nucleus of the cells. Now, in terms of modeling, recent publications have laid out PBPK QSP hybrid approaches for this type of AAV gene therapy, for AAV gene therapy in general. And the PhysioPD platform, in particular, is a hybrid PBPK QSP model. Focuses on the biodistribution and transduction of AAV delivered, but it covers from systematic delivery to local delivery and is defined in a generic way so we can cover different tissues. Now, the following are the processes or features represented in the platform based on our research and previous publications. Some of them are already modeled in previous models or publications, but we added additional features. Now, these are, for example, systemic and localized AAV administration. We also represented plasma flow, the neutralizing antibodies impact, the vascular escape into interstitial space in tissue, the binding to membrane, the AAV binding to membrane receptors and co-receptors, because it depends on the AAV serotype. Co-receptors can be involved in the binding. Then intracytoplasm processes, nuclear internalization, uncoating and second strand synthesis, transgene transcription, and transgene expression. Now we're going to show how the model has been developed, the approach we took for the design and also for the calibration process. This gene therapy platform, as we explained, is a PBPK QSP structure, and we can see here on the right, when we see the overall picture, we see the PBPK structure. We see that with a plasma compartment on the left and on the right, and we see that AAV goes from, can go from plasma to all these different compartments and go back to plasma from all these compartments too. Now, these compartments represent different tissues in our model. The first one represents eye tissue, the second one brain, third one muscle, and the fourth compartment is kind of an average of tissues, peripheral tissues that are not explicitly represented in the model. Now, all these are connected through the planar picture. But now, if we go to the internal structure of a compartment, for example, the compartment 1, all of them right now in the current state of the model, all of them are similar, but they are defined in terms of different parameters. Internally also can be adapted to represent specific characteristics of a tissue. Now, in this compartment, we can see, if we go to the top left, we see that from plasma, we will have AAV coming to the tissue to the right. That is, and in particular to the tissue vasculature that is represented in this compartment at the top, a small compartment, And of course, AAV will leave this tissue vasculature to go back to the plasma to the right. Now, the AAV that remains in the tissue vasculature, it enters the tissue and it goes to the cells that, of course, have a membrane, and the binding of this AAV to receptors is represented in this compartment on the left that represents the membrane of all cells in that tissue. Now, from this compartment, from membrane, AAV goes inside the cell. We have on the right here on the center the compartment that represents cytoplasm of the cell, and then from cytoplasm it will go inside the nucleus on the right, where other processes such as uncoating or mRNA translation take place. Finally, we represent, or at least for qualitative purposes, for human simulations, we have also defined transgene protein being produced and transported to the plasma. Here on this the bottom right, we can see how it goes back. I mean, the transgene protein is produced and is released to plasma. Now, AAV2 is the case study that, or the serotype that we use for the case study. So the platform, in general, as we explained, is flexible. It focuses on AAV-based biodistributions in its current state, and it can cover different species, like, as we mentioned, mouse, non-human primate, and human. And in this particular study, we did the three of them. Now, target tissues, we mostly use, we use data from brain and muscle, not from eye, and we explain why, not for eye tissue. And then we also use data to calibrate the pool compartment, the one that covers the rest of the tissues. Now, AAV vectors, as I mentioned, it's the AAV2, the serotype we focus on. Routes of administration, we have systemic and local, but in this case, we focus on the systemic administration. Now, parameters in the model can be modified to reflect these differences, and we'll show the case as an example of how it can be done. And that makes it very, very versatile for gene therapy research. Now, for the virtual human, we perform qualitative simulations, but it can represent, the platform can represent by scaling from other species or by using human data, of course. We can simulate biodistribution, and we can predict the transgene protein levels on different target tissues. Now, for calibration here, we have summarized the protocols we follow. Now, in this table, we can see the three different columns. We have mouse, NHP, and human. We have more data for mouse in literature than for primates and then human. Now, if we look at the rows instead of the columns, we have on the top short-term data here in pink, and then and this corresponds to extracellular parameters. We can use short-term data to define, like, for example, what happens in the first minute or first 2 minutes, to define parameters or to calibrate parameters that are related to what happens in the beginning of the process, like PBPK values or tissue permeability. Now, we have long-term data that that data can be used to define intracellular parameters, because that happens later in time, like transport rates, clearance rates inside the cell, and other type of rates. So we follow that process. We had some certain data, long-term data, and we started from mouse. So mouse, we were able to calibrate and to define parameters, but also have the data to calibrate what happens short-term and long-term. And from there, we made some assumptions for NHP, mostly similar intracellular parameters across species. But then we use NHP data for some of the extracellular parameters. And finally, for human, we scale. I mean, PBPK values obviously are different for humans than other species. So we took from literature, and other parameters we scale from NHP, and other parameters we assume were similar across species. Now, in terms of simulations, we'll start with the mouse simulations. For the mouse simulation, we took this specific study from 2016. Now, we were focusing on a case study that was very specific, so we needed to find data that we could use even if we didn't have the whole picture. In this particular case, in this study, they used AAV2, so that fitted with what we wanted. The administration of the dose was intravenous administration. It was tail vein injection. Then the data that they reported, what they measure is like vector genome per microgram of DNA in brain, in muscle, and in other tissues. Unfortunately, they didn't have data for eye, but at least it was a very good paper to calibrate the compartments we were trying to define and represent for this study. Now, in terms of data, also they had two key time points. They had short-term data, 2 minutes data, post-administration data. So it was some data about blood concentration in blood of AAV and biodistribution, AAV concentrations in different tissues. And then 14 days, some data about what happened 2 days after the dose was administered. Now here we can see on the left the mouse data, and on the right our simulations with the model. The mouse data showed around 33% of the concentration of AAV in blood 2 minutes after administration. Now, on the right, we see how, starting from 100% concentration in blood, it decreases over time fast in the first minute, and then after 2 minutes, we reach the 33% concentration that is expected according to the data. Now, at the bottom, we see the concentrations, AAV concentration in different tissues. In pink, we have brain. In light blue, we have the peripheral tissues. In dark blue, we have muscle. Those colors correspond to our simulations on the right. We represented the data in green for the particular at 2 minutes. And the pooled compartment, the light blue, that represents the peripheral tissues, for that one, because we have different concentrations that are different, particularly in order of magnitude, we define a geometric mean across all tissues to define basically the data that we were trying to fit with our model. Now, long-term data was, as I mentioned, it was information in what happens at 2 weeks. The data is on the left. The concentrations decrease in all tissues. And again, for the pool compartment, we needed to use a geometric mean, but we were able to capture both in short-term and long-term the concentrations that were provided by this publication. So as we can see, it was very important to distinguish between what happens short-term and long-term, because the dynamic is different. We can see short-term things decrease fast in terms of minutes, and then things are quite stable after that, and for the long-term behavior. Now, to create a virtual NHP, we also we took a different reference, this one from 2006, and This study the goal of this study was to study the biodistribution of AAV2 using IV injection in cynos in these primates. Now, because it was AAV2, IV administration, they reported transduction efficiency for relevant tissues, like except for eye, again. They reported a vector genome per mass of DNA. That was relevant for us too, and didn't have short-term data, but they had long-term data. In this case, even longer than weeks. It was 3 months post-administration. Now, the NHP data of this paper is here on the left. We have a long list of tissues. We have 4 monkeys, 4 columns, 1 per monkey. We didn't have much data. Many samples. We only have 4 samples to look at. But this was good enough because they provided the symbols in the table. These minus and plus symbols represent different ranges of concentrations of genome per vector genome per mass of DNA. We can see these ranges here on the second item on the right. The more symbols, the more concentration. But we can see these are ranges that cover one range of one order of magnitude. So the differences can be high when you compare different tissues. Now, they had explicit, they had at least a concentration, a range of concentrations for the tissues, specific tissues we were representing, brain and muscle. And then we had to take kind of a geometric mean across all tissues because we can see some of them in the table, like a spleen, have high concentrations of AAV, but there are other tissues that don't have, didn't get any AAV, or other tissues. So we have the simulations on the bottom right figure. The empty circles in the plot around the solid circles represent all these 4 primates, and then the solid circles represent the geometric mean across primates for each tissue. And we can see we were able also to calibrate parameters and fit this data properly. Finally, for a human, we decided to implement or to use more parts of the model, because so far we have focused on biodistribution. We didn't assume any effect of neutralizing antibodies in plasma or tissue, but in this case, for human, we added that. We added the neutralizing antibodies, and we also analyze what happens with transgene protein in plasma. Now, on the left, we can see, as I mentioned, neutralizing antibodies. On the very left, basically, the first rectangle, we have what happens in plasma. We have neutralizing antibodies, but also we have the dosing there, the systemic administration. The other rectangle on the right with the 1 is local administration and also neutralizing antibodies acting locally. Then the square in the center, the pink square with the number 2 represents the protein translation in the cytoplasm and protein secretion from cytoplasm to tissue. In this first study, the payload protein was assumed to be a secreted protein. Now, for a protein transport between tissue and plasma, protein production and degradation, protein transport between tissue and plasma, and degradation in plasma, we have what we included on the right of the diagram. Now, for qualitative human results, we just show with these results the predicted ability of the platform. We didn't compare to data. We just use qualitative comparison. First, on the top right, we see as we represented for the other species. We don't have data, so we just see how the concentrations will evolve for a human after applying proper PBPK values and some allometric scaling. And then the bottom, we can see at the bottom left, we can see some data, the results of simulations from a paper that was written by Rao. The reference of this web paper or chapter of a book that was from two years ago, 2024. And this model that has been one of our main references for our model, they simulated, they were using a different, I mean, this data actually is from factor 9 activity in plasma. This paper and this data was about AAV8, not AAV2 serotype. And also the target tissue was liver. So it's a different study, different goal in the project, but we wanted to compare by using similar parameters or similar order of magnitudes of parameters that they use for to define or to predict these protein levels in plasma. We use similar parameters in our model, we implemented, and we got qualitatively, we got similar dynamic, and we represent here different doses. The higher the dose, the higher the higher the AAV dose, the higher the transgene protein concentration in plasma. Finally, for conclusions, we can summarize by saying that Rosa has developed a versatile biodistribution and transduction model for gene therapy research, a platform that can simulate different biodistribution scenarios, and again, perform transgene protein analysis. It can predict protein distribution in different species, including humans. So this is a platform that is versatile in terms of species. If we have more data about mouse or other species, we can use that and expand the platform from there to humans. And it can be adapted and extended to include local delivery to inaccessible tissues, such as the ocular space, and other routes of administration, such as intravitreal delivery, that will be very useful for the eye compartment. Now, parameters can be modified to reflect, this is again a summary of the things we can modify in the model by changing parameters. For example, in terms of local and systemic routes of administration, we can parameterize the compartments depending on the tissues of interest. We can use AAV, but also LNP vector can be also implemented in the model. We can deliver, the parameters can be modified to represent a specific payload. And the model can also assess the biodistribution of non-native AAVs, such as engineered capsids. So thank you for attending the webinar. Please do not hesitate to ask any question. Thank you again.
Thank you, Alvaro. That was a really nice presentation. We do have a couple of questions from the audience. First question is, did you assume that AAV binding affinity was the same in all of the tissues that are represented in the model?
No, we define different binding rates, for different tissues, as far as I remember.
Okay. Next question is, what would be the process for adapting the model to represent LNP or other vectors? Like, what would be different in the model, and what kind of data might you need to define the parameters?
So, well, we know that obviously the characteristics are different. I mean, some mechanisms are different. Like, for example, there's viral vectors trigger immune responses. Well, LNP, for example, does not trigger. Or non-viral vectors in general, right? So those are factors that we can define in terms of parameters. We can define different parameters just to distinguish between, the difference in vulnerability of the vector to be, administer in a specific to a specific tissue. There are other things like for example, LNPs have more they are more vulnerable, even if they are more vulnerable to intracellular and extracellular barriers. So,
Hmm.
so again, obviously we will need to change parameter values like we do across tissues, right, or even across species.
Right.
That's for sure. Some mechanisms that are different. We obviously we need to modify partially the layout of the model
Hmm.
internally in the tissue mostly, but yeah, we can do that. That's why it makes it versatile because the structure of the model, the parameters obviously change, but the structure of the
Hmm.
model is quite general
Yeah.
and includes, yeah, as we mentioned, like different ways of administering the drug and all that. So LNP, we could adapt to that. Yeah.
Okay.
Mhm.
You discussed another question. You discussed using the geometric mean to derive expected values for the quote unquote other compartment concentration.
Mhm.
How did you decide between this data transformation approach versus representing additional tissues explicitly in the model based on
Yeah.
species specific information or available data sets? And then the second part of this question is the number of tissue types represented in the platform model configurable or parameterizable? So I think, can you
Uh,
adapt the model for other compartments that represent different tissues?
Sure, yes. The thing is that the model, this first version of the model, the original one, it's adapted for this particular case that we
Hmm.
were interested in. This
Yeah.
AAV2 vector, and we were focusing on three specific tissues, although we only use data for two to calibrate two. We still have the eye compartment. And so the rest of the tissues, we didn't need like a specific, I mean, we didn't need to get into details and with the other tissues. So for simplicity, we pulled them into one compartment. And because we saw in both studies, in mouse and in monkey, we saw that the order of magnitude was quite different across tissues, like for example, lung in the mouse case compared to other tissues, or in the monkey cases also we had also differences in not only in terms of across tissues, but also, for example, for the monkey case across monkeys. We only had four monkeys, but some of them had much higher concentrations of AAV
Hmm.
than other monkeys. So we consider that geometric mean because you have this wide range of values with different orders of magnitude. A geometric mean was more reasonable than an arithmetic mean. And so for the other tissues in particular, because that was a huge difference across tissues. Yeah.
Hmm. I guess one other comment I can make is that we also consider the timeliness and the expense of creating a simple model versus creating a more complex model where there's diminishing returns representing tissues where we don't believe there's very much distribution of the vector. So there are some trade-offs. We could represent every tissue explicitly, but maybe it's not that much benefit compared to the additional time it would take to add compartments and parameterize them. So that's one of the considerations in the model scope.
Yeah.
Next question is, how do you build confidence in the model parameters, especially the human model, human parameterization?
Yeah. Yeah, so obviously it's tricky because for human in this particular case also we didn't have data that we could use. So that's why the calibration protocol that we follow it can be improved and it can be more accurate. Obviously, again depending on the data you have, and short term and long term, as I mentioned in this slide where I summarize the protocol of that we follow, we had data across species. We have more data for mouse that are obviously very different to human, but then we have the primates in between. So we kind of started from mouse because we had more data, and we had short-term data, long-term data to distinguish between what happens in the first minutes and then what happens weeks or months after that. So that confidence, obviously the more data you have, maybe the clearer the picture. But at least with this protocol, we were able to have quite high confidence for the parameters we were defining with the species we had more data about, and also to distinguish what the shorter data was important for some set of parameters, while the long-term data was more important for other set of parameters because things happen later in time, right? Inside the membrane, inside the cells and all that. I mean, inside the cells. So again, to build that confidence, you can define like different stages depending on the data. You build from there. You have to make some assumptions, obviously, for some parameters. When you move to the next species, if you don't have that much data, and we did for primate to transition from mouse to primate, we didn't have to make many assumptions. Fortunately, we had enough data to calibrate a primate. But then for human, yeah, that's the trickiest part. We made assumptions like about intracellular parameters, considering that inside the cell across species, more or less the same dynamic, and then out extracellular parameters like they are mostly like PBPK values or the vascular, the binding rates may change too, but mostly PBPK values, number of cells per tissue that we can consider that where the AAV is targeting. So those we had literature, a lot of literature for that. So because we had that and we made some assumptions that obviously can be refined and polished in the more data we have from human, obviously that's something that we could do in a next step. But that as a first, connection across species, I mean, we consider the assumptions we made were reasonable, but it's true that we will need to check or to validate the model, or using some data and test how accurate our predictions are, for sure. That would be our next step.
Okay, just a comment that we can continue up until the top of the hour. We have a lot of questions, so if people are willing to hang in there, we've got a few more questions. And I'll also ask the speaker, are you okay hanging out until the top of the hour to answer questions?
Sure, for sure.
Okay, great. So the next question is, did you integrate virtual populations to evaluate the impact of variability? And if so, how did you define them? And if you didn't do it for this project, how would you do that in the future? Virtual populations,
sure. Virtual populations, sure. Yeah, we didn't do that in this project. We didn't get to that point. But sure, the variability, we will define, I mean, different parameters, for example, right? We will need to define different, we have ranges of values of binding rates. In fact, we had some of the parameters we defined, they were defined in terms of ranges, and they were calibrated to fit this data from those studies. But obviously, we can generate, like we did for these particular cases, they were like kind of virtual mouse, virtual NHP that fitted a specific set of data that can change, generate like a different set of parameters, always defined in ranges that are physiologically realistic, and generate this population to see how small changes in these parameters could give us different responses. Yeah, and different biodistribution in particular, different across tissues.
Okay. The next question is more of a comment, but maybe we can speculate on this as well. There has been some reports in the literature about the toxic effects of large doses or large concentrations of LNPs. And are there other approaches to gene delivery that might be less toxic? And for example, extracellular vesicles that don't incorporate in the cell membrane. And I don't know if you have background in looking at this, but can you speculate on that? And if not, maybe we can follow up offline after this.
Okay. Well, if it's in terms of toxicity, I mean, the question is about that.
This
And
is
it's
more about
true,
the
it's true
vector
that.
and not the gene you're delivering.
Mhm. Sure, I will need to look into that because it's not something we really we didn't go into detail on that. But for sure, like we could adapt that to this model. I mean, we will need to
Mhm.
see how much it needs to be modified, and also depending on the tissue, I guess there is variability there too. But yeah, I mean, maybe I can answer that question via email because I will need to look
Yeah.
into that.
Okay, we'll follow up. Okay, next question. Depending on the tissues and the rates of cell division, AAVs might clear at different rates, different tissues. Are there any mechanisms or parameters that account for clearance that is specific within a specific tissue? This could help predict duration of efficacy. You know, if something is cleared more quickly or more slowly in the site of action versus the non-desired site that you get distribution.
Sure. We have that defined explicitly, like how fast it clears. And again, because we split the model into different compartments, you can define different clearance rates depending on the tissue and the situation you are studying. So for sure, we can implement that easily. It's already in the
Mhm.
model. Mhm.
Okay, next question, and I think you've alluded to this earlier. Did you face challenges with the identifiability of key or sensitive parameters? And if parameters weren't easily identifiable, how did you evaluate the sensitivity of that uncertainty?
Okay, we didn't perform sensitivity analysis like, I mean, we actually. I'm trying to remember. But in any case, yes, there were parameters that were challenging to get. As we mentioned in the beginning of the presentation, we did research about papers, models, PBPK or PBPK QSP models that have been published. And there were parameters, like mostly what happens inside the cell, that like encoding rates or these type of rates, the transition from DNA to mRNA, it wasn't. In fact, actually, for mouse and primate, we didn't need to define what happened after. Basically, we have the double-stranded DNA. After that, is mostly it was a qualitative analysis for human. But because of the lack of data, that was we used this other reference. They had some data, and we made a qualitative comparison. So basically, I would say before what happens in the nucleus, or right when the AAV gets into the nucleus of the cell, we were quite confident that we had some
Oh, Alvaro, we're losing your audio.
for the.
Can you
Oh,
repeat the answer to that
sorry.
question? We lost your audio there.
Let me. Yeah, I think there is a delay. I don't know if I can reconnect to avoid the delay.
No,
If it,
that's okay. I would just go with what you're connected to.
okay. So basically, what I was saying is that in the model, the way we proceeded, we had the rates, the parameters that we defined before we had I mean the whole protein expression, right? The transgene protein and all that. For mouse and primate, we didn't need those rates. We didn't need those parameters specifically. So the uncertainty of that wasn't a problem for these cases. Although there were rates like binding rates or encoding rates that were challenging to find, and in some cases we had to calibrate. Now, after that, in particular in the case we were studying, like basically when you have mRNA, transgene protein, and all that, that part, we looked into some sources and it wasn't easy to find some of these rates. I mean, we saw that we were calibrated also by other authors to fit some data, and the data we found published, it was an AAV2 in human, it was in other serotype, and it was a concentration of the in plasma of the transgene protein. So that, again, we did it in terms of qualitative purposes because we saw that we needed more data and we needed to, I mean, we'll get into that point, but we didn't do it in this project because of the high uncertainty. Yeah.
Okay. Next question is, how were the translation rates calibrated? Was it assumed to be the same across all species? Because there is some literature to suggest that the translation rate can vary across different species. And I'm not sure if this question refers to the translation of the gene product. Yes.
Yeah, again, the last rate that we had to calibrate, it was the encoding rate. I don't remember right now, we did define different values, but again, mostly what we had to calibrate the binding rates and the encoding rates. Others were kind of, we found in the literature what happens in the cytoplasm, the transition to nucleus and all that. So we didn't have to deal with that for this particular case, as far as I remember.
Yeah, and just a comment that the focus in this project was on the distribution, not on the efficacy. So we didn't do much in the direction of the amount of gene product and its effect on the disease indication. But the model can certainly be adapted to do that. But one of the considerations that the questioner asked was translation of this across species. Good question. Okay, next question. Did you consider including in the model mechanisms to inactivate the donor DNA?
No, we didn't consider that in the model, during this project. Yeah.
Next question, is the platform limited to preclinical and human adults, or could you also use it for special populations? For example, can you predict the durability of liver-targeted gene therapy in children whose liver size may be different, both because of their age, but also maybe the disease state in terms of hepatocytes proliferating, for example?
Well, that's an interesting question, and yeah, I mean, this was well, again, this was for calibrated for animals, actually, not for the human. But sure, because we have all these in terms of extracellular parameters that are related to, as I mentioned, like the PBPK values, right? The different flow rates, the different size of the tissues or the organs. But also the number of cells per tissue, all that. Sure, like we could adapt that to a different type of population, like a child instead of an adult, for sure. Yeah.
Next question, how is the AAV concentration determined within the cellular compartments? For example, membrane, cytoplasm, nucleus, and across different species as shown in the model diagram. So I think this is more of a data question.
Oh, yeah. The way we proceeded is we sum up the concentrations from the concentration in tissue extra, I mean outside the tissue, or in the interstitial space, and then inside the cell. So we assume that the concentration, the AAV concentration, or in this case, the data was a vector genome per mass of DNA. So we took the whole, I mean, concentration from the concentration that it gets to the tissue vasculature to the AAV concentration that is inside the cell. So we assume it was the total amount.
Yeah, I think there was also in vitro data to characterize the intracellular distribution of the vector as well. Okay, next question.
Yeah.
how many parameters were fixed across the species, and what were they, and why did we decide to fix some and not others? And what other parameters were calibrated with mouse data and monkey data separately? And this is, I think you can probably answer this in a more general way rather than naming every parameter, but the gist of it is, how did we decide to fix some and adapt others?
Mhm. Yeah, I mentioned earlier that we had in the table also they distinguish between extracellular parameters and intracellular parameters. So, and again, we kind of realized that short-term data, we could fit better short-term data with the first parameters, with the one connected to extracellular behavior, because obviously it's too fast and nothing really happens in the cells. I mean, it happens, but it's not as determinant as, for example, the PBPK values. So we define some parameters. I mean, for mouse, we started from scratch and we had data about the extracellular parameters. I think we had to define well, I'm not gonna get into detail parameter as you said, but sure, we had in extracellular parameters we found in literature. Now, intracellular parameters we found some parameters, but as I mentioned, the good thing about starting from mouse is that we had the data, so. And we could, because we split the data into two parts, we could kind of connect the binding rates values with the short-term data and some tissue permeability, for example, that was an important factor too, the tissue permeability that happens in the beginning. So those factors, the short-term data help us with those. What happened inside the cell, the long-term data for mouse was really useful to define that, like coating rates and that. Others we found in literature, like transport rates or clearance rate. We could estimate by obviously making some assumptions and knowing some information about how AAV can be transported inside a cell. But then when you jump to the next species, yeah, you have to make some assumptions. Monkey, we didn't have that much. And one important assumption that we made that, again, it can be refined with more data if it's necessary, is that we, after doing research, the proper research, we realized that we could assume, at least for now, that, and for these purposes, for this project, that intracellular parameters were kind of similar across the species. That simplified a lot for the next steps, right? To go to a primate and to go to human. Now, the data we had then for primate, for example, helped to define what was still different. And PBPK and other values were from literature for primate anyway. So not for this particular study, but obviously we know the physiology, the size of the tissues and all that in a primate. So it was a combination of different things, like research and assumptions and some data that we needed to calibrate. Yeah.
Great. Next question. How many parameters? Sorry, we just answered that one. Next question is, did you encounter challenges converting between units? So, expression of DNA being expressed per microgram of total DNA or versus amount of mass in the genome. How do you handle this when mapping across units between species and different types of samples that are taken, blood versus tissue?
Mm-hmm. That's an interesting question. I, I will need to remember because that's getting into details about how we dealt with the units. I remember we had data, for example, the data with this provided there, that was provided by these two studies was like the vector genome per mass of DNA. So we had we had to make use some data about the mass of DNA that we had per species or per tissue. I think it was per tissue. And that was an important factor to be able to compare different I mean, to compare in our results with the data. Now, in terms of units, I remember a good, I mean, there was this good reference that we mentioned that we use partially for to develop our model, this paper or chapter in a book from Rao. In that one, we kind of follow the same protocol that they did in terms of units, where to define the vector genome since the beginning. And obviously, we had to consider different depending on the data that we were getting, we were using. If it was, for example, concentration, we had to take into account the tissue volume or again, the number of cells, for example, for the total mass of DNA in the entire tissue. I mean, we had to make some transformations. I mean, I don't know if I have to get into detail of how we did for every
No,
particular part of the,
yeah, we can just
but
comment.
yeah,
This is a
we
challenge
play
in
with
modeling in
in the
general.
transformation.
Yeah, this is a challenge in modeling in general when you're going across different
Yeah.
tissues and the way things are measured as concentrations, depending on the matrix you're assaying.
Mhm.
Last question here. Did you have long and short-term pharmacodynamic readouts, primarily in mouse or in mouse and non-human primates? And how were the time points decided?
Sorry, could you repeat the beginning of the question? I mean,
Did we have long or short-term pharmacodynamic readouts, primarily in mouse or in non-human primates? And how were the time points decided for simulation and for measurement? And just a quick comment for the questioner. Rosa did the modeling. We didn't do any of the data collection in the mice and monkeys.
yeah, exactly. Thank you, Mike. I mean, this is the thing. We were focusing on a specific case so we needed paper, we needed studies that at least met some criteria, like the type of AAV, like the tissues that we were targeting, because AAV2 is used frequently for eye tissue, but finding a paper that where it was administered intravenously, this AAV2 targeting these specific tissues, or at least that it had this information about brain and muscle. So we had to reduce our options to just to few papers like these ones. And they provided the data they provided, like a specific time point. So we just used that to the best way we could to calibrate the model. Yeah.
Thank you, Alvaro.
But not PK as far as I remember. No.
Okay. Well, I'd like to thank the speaker, Alvaro Ruiz-Martinez, and I'd like to thank the attendees for their excellent questions. That was a very productive webinar session. And just to remind the audience that our next speaker is coming up on February 11th, Emily Nieves, and we look forward to seeing you then. Thanks, everybody, and have a good rest of your week.
Mhm.